Hi, good afternoon, everyone. I'm Ravi Kalatur, Senior Field Application Scientist for QDI Solutions. The title of my presentation is AI-powered hereditary disease diagnostics and how we can use this to close the gap in clinical exome completeness. Before I go into my presentation, I would like to thank all the participants who are attending this seminar. And I would also like to thank the organizers for providing me this opportunity to present our clinical decision software, which is QCI Interpret. Just as a disclaimer, the QCI Interpret is an evidence-based decision support software intended as an aid to interpret of variants observed in genomics next-generation sequencing data. The software is not intended as a primary diagnostic tool by physicians or to be used as a substitute for professional healthcare advice. The agenda of today's presentation is to understand the current landscape of hereditary disease testing, to provide a brief introduction to QDI and our variant interpretation tool, QCI Interpret, and talk about how we are using AI-powered hereditary disease diagnosis combined with our manually curated content to accurately interpret variants obtained after a secondary analysis. In ancient Greek medicines, the Hippocrates theory of four humors is black bile, yellow bile, and blood and phlegm. And these four humors are used to determine the health condition of an individual. Currently, we moved from those four humors to four nucleotides. And these four nucleotides determine our health and disease status. Hereditary NGS testing and interpretation plays a crucial role at every stage of our life. That is from zygote to adult. The NIPT testing is quite important during pregnancy, NICU diagnostics, and Mendelian disorders during infancy, and during adulthood to diagnose with hereditary cancers or somatic cancer, and during the aging process, we can use the pharmacogenomics to determine the health condition of an individual. The hereditary testing field has constantly been evolving over the time, which started as a single gene diagnosis in 2000. It has moved to multi-gene hotspot panel in 2010. And between 2020 and 2025, we have multiple gene exon panels and whole exome and whole genome panels coming into prominence. And a decade later, we might use multi-omics approach in order to test human diseases. Currently, we are at multi-gene exome panel and whole genome and whole exome panel. Diagnosis to detect if there are any diseased conditions present in an individual. There is a constant growth in genotype and phenotype relationships. There is an increase in phenotypes with known molecular diseases and also with genes with phenotype causing mutations. Currently, according to WOMIM morbid map, we have about 4,481 genes that has disorders and traits. And every new gene has been identified per day, and it is associated with hereditary disorder phenotypes. Overall, it is estimated that there are about 9,000 genes which are involved in hereditary diseases. So we have about 4,000 genes are yet to be identified, which are linked to hereditary disease. With over 25,000 variants in whole exome and more than 3 million variants observed in whole genome, how can one confidently and quickly analyze and interpret these variants from your NGS tests? And there are three key challenges for clinical labs conducting NGS testing. One is they need a fast interpretation, which means that they need to diagnose the patient's disease in a very quick time. And it can be very challenging when it comes to large gene panels and whole exome or whole genome data to analyze them and to interpret those variants in a quick time. Additionally, in order to develop these secondary analysis and tertiary analysis variant interpretation tools could be quite costly. And these labs can invest in identifying the variants, their time in identifying the variants rather than developing the tools. Additionally, it is also quite labor intensive to keep the variant databases and content up to date. And there are several, with the new information coming every day, it is necessary to keep the information up to date in order to call the variants that are identified in a human sample to accurately predict or interpret those variants. Additionally, there are other challenges apart from the three challenges that I mentioned in previous slide. The fact is that the current trend is the large panel approach, and which is being transitioned to whole exome and whole genome sequencing. With this huge amount of data, the challenges are we identify about thousands to millions of variants in this sample. Interpretation time is critical. And there is an increased demand of new evidence for accurate variant interpretation. Need for high quality curated data to use for high quality interpretation. And this can be partly solved by combination of artificial intelligence and manually curated data. And we can combine them in order to efficiently interpret these variants. Keeping in this mind, we will move to our next section of the presentation, which is, I'll give you a brief introduction to QIAGEN Digital Insights and QCI Interpret software. Our QCI Interpret software is leading provider for genomic and clinical knowledge. And we have about 200 scientists and several software engineers who are involved in developing QCI Interpret software. And this helps us to provide a valuable tool for scientists and clinicians who can use this tool to interpret your variants accurately and correctly. Our curation effort led to 21 million curated scientific findings, and we have about 90,000 satisfied customers worldwide. And QCI Interpret software has been cited in more than 35,000 publications. And we have interpreted over 3 million patient samples. What makes QIAGEN the preferred partner in variant interpretation? Behind the QCI software, we have an industry-leading clinical knowledge base. And this knowledge base is based upon 5 pillars. These pillars are depth of the information that we curate, breadth, quality, timeliness, and context. As I mentioned before, we have more than 21 million curated findings, about 9 million variants, and we use 40 plus public databases in order to help our customers in their variant interpretation process. And we have been doing this for past 21 years. Additionally, we have added the AI-driven insights on top of our manually curated data in order to interpret those rare disease variants. And our knowledge base is updated weekly, and about 5,000 plus variants are added daily. And we integrate this information into knowledge base using a unified ontology. Our knowledge base foundation is based upon current contents and external databases. The curated content comes from clinical trials, FDA, HGMD, which is a priority software for us, which is uniquely licensed to QCI. And our classification is based upon ACMG, AMP, and ASCO guidelines. Apart from this, we also integrate information coming from external databases such as DBSNP, 1000 Genomes, CleanVar, Cosmic, etc. And we integrate both manually curated content and the information coming from external databases into specified ontologies such as patient phenotype, disease mechanism, cellular mechanism, so on and so forth. With this, we have currently more than 9 million variants in our knowledge base. The QCI Interpret is a flexible and scalable evidence-based variant interpretation platform. Here in QIAGEN, we have a complete solution starting from sequencing data all the way to reporting of the variants. And first of all, after performing a wet lab experiment, we perform the sequencing and obtain FASTQ files. These FASTQ files are then passed on to secondary analysis tool, which we have, which is CLC Genomics Workbench, which is platform agnostic. It takes sequencing files or FASTQ files coming from any platform, short read, long read, Illumina, Oxford Nanopore, Antorrent, MGI, BGI, etc. Additionally, we also developed a world fastest algorithm, which takes the FASTQ files and generates VCF files in very quick time. Once the VCF file is generated, you can upload this VCF file into our QCI Interpret software for variant interpretation. We have set of filtering options that can be used to classify the variants. And with our knowledge base, once we have classified and interpret the variants confidently, you can submit these variants for a report as a PDF or a text file. And you can connect through your own LIMS system using our APIs, which can help you to integrate the knowledge coming or the variant interpretation happening in our interpretation software directly into your LIMS system. The QCI interpret software is powered by the knowledge base, as I mentioned before. It provides interactive filters to prioritize the variants. And the variant classification is done on ACMG AMP guidelines. Additionally, we also have a high-quality phenotype-driven ranking for prioritization. And this prioritization helps quickly to interpret your variants or rank your variants for easy interpretation. And it also generates evidence-based reports with efficiency, confidence, and reproducibility. Currently, we are achieving our wide IVDR affiliation. And with this, we have annotated about 3 million patient cases worldwide. What we can do with QCI Interpret software? We can diagnose hereditary cancer. We can perform carrier screening. We can do trio analysis, whole genome, whole exome variant interpretation, or interpretation of hereditary disorders or somatic oncology. How we do? We do with the QCI Interpret software is the only interpretation platform which contains most extensive created evidence and content of the literature to ensure high quality interpretation combined with AI-driven content. The unique feature, in order to improve interpretation within hereditary disease, we have a priority interactive filter cascade, which is based upon several filtering options, such as confidence filters, which is based upon the quality. You can remove variants that are present in common population. We can filter variants based upon predicted deleterious and genetic analysis. What these filtering cascades helps to do is to rapidly identify clinically relevant variants, quickly narrow down the lists of variants down through an interactive series of filters. And additionally, we also recently added a QCI Interpret copy number variation interpretation to our system. Currently, we have about 60,000 HGMD, QIAGEN curated CNV literature references. It is based upon new ACMG CNV guidelines, computed pathogenicity classification, and it also links to external CNV databases and nomad CNV frequencies, which will help our user to efficiently classify CNV pathogenicity. The second unique feature is phenotype-driven ranking. The phenotype-driven ranking algorithm ranks and prioritizes candidate diseases. This can changes and prioritizes variants dynamically, and it can be added to hereditary analysis at any time during the analysis. And this phenotype-driven ranking is supported by clinical validity. In the clinical validity, the gene and disease association strengths are computed for all the genes. And candidate disease evidence diagrams and findings and literature references are available for review. And this is based upon QIAGEN's framework. Additionally, we also integrated a triage mode that can really help the various assessment can be made directly on the variant list page when screening many variants in a given case. Apart from this, we also have optional columns enabled via settings for quick determination of sample quality, such as information on the position of the variant, links to ClinVar, and also literature, which can help in faster assessment of variants. Additionally, a user can also add secondary reports that are coming from clinical labs to the particular sample, such as cytogenetic analysis report or a medical report coming from the lab for that particular patient, and also quality information coming from your secondary analysis. This can help you to determine the variants that you have identified in the patients after sequencing. By using all this information, you can generate a customizable report with a standardized interpretation, as shown in the slide here. Now we move on to the last part, which is AI-powered hereditary diagnosis and how we combine this with manually curated content. As I mentioned before, we have about 200 PhD, MD level scientists who go through publications and manually curate the information which is published. And recently, we have added AI-driven literature extraction for thousands of rare disease genes in order to assess the variants, especially variants of unknown significance. And we have developed a new AI-trained phenotype-driven ranking in order to prioritize the candidate variants. And these new features will help to reduce the time needed for prioritization, assessment, and interpretation of clinically relevant variants. The AI content is on top of manually curated data. So the manually curated data is the basis for our variant interpretation software, which is QCI1. And we have the full transparency on the articles and the source of content. And this enhanced content and list of variants and literature from natural language processing exists on top of the manually curated content. And this helps to reduce turnaround time, less clicks and less time on screen, new gene variant and disease phenotype information with new disease filtering capabilities helps in efficient variant interpretation of whole genome and whole exome data. As I mentioned before, we have integrated AI-driven bibliography content. And this is represented, as I shown here, with chip symbols. The blue chip means AI intelligence identified the references as mentioned with high confidence, whereas the orange chip means medium and high medium confidence. We don't integrate low confidence research articles in our database. And this is on top of, this is additional information on top of our manually curated content, which is core to our QCI interpret software. And expanding the bibliographic coverage of exome complete coverage of rare inherited or rare hereditary disease is obtained through a combination of AI-driven bibliography with existing state-of-art manually curated bibliography. And what this does is it helps for faster and accurate diagnosis. Additionally, you can filter this bibliography based upon phenotype-related references only. So I would like to show you how a phenotype-driven ranking can help to prioritize your variants in next few slides. Say, for example, in one of the files here, you can see there are about 999 variants that we see coming from this VCF file. And these many variants are slightly difficult in order to interpret them efficiently. So with our aid of our strong filtering steps and phenotype-driven ranking, where you can type in the phenotypes of the patients. Say, for example, here we have 3 phenotypes, such as congenital erythroderma, disorder of hair, scaly erythemia. And once with this patient phenotype, you can filter, and in combination with our filter settings, you can filter down these variants to 188. And here you can see that SPINK-K5 can be prioritized and can be a candidate for Netherton syndrome. And there are 2 variants of different classification coming from the same gene, which is SPINK5. And once you click on the phenotype-driven ranking, we can provide a network, phenotype network, that links gene with the disease, and then the gene is colored in orange, the disease is colored in green, and the phenotypes that we have entered are colored in blue here. And by clicking on these edges between these nodes, you get the findings or links to the literature from where this relationship has been obtained from. Additionally, this information can be obtained from View Bibliography option. Once you click on View Bibliography option and filter based upon Show Phenotype Related References only, it helps to filter out the variants which are highly specific and they're related to this particular phenotype. And here you can see that these references are obtained by our powerful AI curated information. Additionally, identifying or interpreting variant of unknown significance is a big challenge currently. And this VUS comes in large numbers when you are analyzing whole genome or whole exome data. And here we have identified ATP2B3 as an variant of unknown significance in this VCF file. And we have used our literature in order to see if there is any information available so that it can help us to interpret this variant. So our powerful AI-driven phenotype and bibliography helped us to identify one particular publication which is published last year, and where they have shown this particular variant is mentioned in this particular reference. And this can be used to help in classifying this particular variant. To end, our QCI Interpret software helps us to analyze the data with precision and confidence. It is scalable, it is standardized, and streamlined for NGS interpretation and reporting workflow. It can help to interpret all types of genetic variations, including CNVs. There is an efficient variant filtering options, and AI-driven phenotype-driven ranking helps to prioritize your variants. And with a unique combination of AI and manually curated content for the best-in-class clinical evidences and faster interpretations. All this will help to reduce interpretation time and not only reduce interpretation time, but also interpret your variants confidently and accurately. With this, I would like to end my presentations. Thank you very much for attending this conference.